visiting my poster. My name is Andrew Patt, and I'm a bioinformatics scientist at the National Center for Advancing Translational Sciences. Today I'm going to talk about better approaches for functional interpretation of metabolite data. Metabolites exhibit a close relationship with phenotype and outcomes. However, properties of the metabolomics data have hindered the impact of this data type in bioinformatics research. To address this, we developed two complementary tools, RaMP-DB and MetaboSPAN. RaMP-DB is a metadatabase that harmonizes annotations from multiple open-source databases, including HMDB, KEGG, Reactome, and Wikipathways. Information types included in RaMP-DB include pathways, ontologies, and chemical classes and structures. A major focus of recent updates has been to include a wider breadth of lipid annotations to address the needs of the lipidomics research community. RaMP-DB is accessible by computational and non-computational users alike using our web interface, API, and R package. Despite RaMP's usefulness in providing comprehensive functional annotations for multi-omic data, there are still some issues with metabolomics data interpretation that we felt weren't addressed by current pathway analysis methodologies. For example, anyone who's worked with mass spec or NMR technologies knows that it's impossible to consistently detect all metabolites in a biospecimen with a single assay, leading to incomplete and inconsistent measurements. Even if these features are detected, it's often difficult to correctly identify them due to gaps in spectral databases. Lastly, many metabolites don't have functional annotations associated with them. To tackle these issues, we propose a similarity network-based approach. We constructed a variety of metabolite similarity network models that describe functional overlap between metabolites within RaMP-DB. As one would expect, these networks are highly modular with tight clusters of functionally related metabolites. In these graphs, nodes represent metabolites while edges are weighted and encode the shared annotations between adjacent metabolites. For example, in our pathway similarity graphs, edges are weighted by an overlap index describing shared pathways between two metabolites. Like for pathway analysis in RaMP, we constructed these network models for lists of biospecimens stored in RaMP, as well as for all metabolites in RaMP. We used these knowledge graphs to increase the scope of pathway analyses in metabolite data in order to reduce the reliance on consistent detection and identification. To do this, we overlaid metabolites of interest onto our knowledge graphs and used network topological properties to extract relevant neighborhoods of metabolites that were highly proximal to the input set. The network topology was described using a Markov random walk with restarts, which allowed us to correct for bias introduced by network connectivity and centrality. We performed a variety of validation experiments with real and simulated data to test the efficacy of MetaboSPAN. In testing, we compared MetaboSPAN's performance against several state-of-the-art pathway analysis methods, taking care to use the same pathway library with each method. First, we tested MetaboSPAN on data in which metabolite labels were randomized to simulate data with no signal. We found that MetaboSPAN exhibited comparable false positive rates to other methods. Next, we generated simulated significant metabolite lists that comprised 50% of the metabolites in randomly selected pathways, which were defined as true positives. We tracked the recovery of these pathways, as well as pathways outside of this list. We found the MetaboSPAN increased maximum true positive recall and decreased medium false positive recall relative to Fisher's exact test, which is the test underlying MetaboSPAN. Next, we tested the overlap of pathway results from datasets generated from one cohort on separate platforms. MetaboSPAN improved the agreement between results over all but one of the competing methods. Lastly, we applied MetaboSPAN to a real dataset generated by our group in a study comparing the metabolomes of dedifferentiated liposarcoma cell lines stratified by their MDM2 copy numbers. We compared the results of MetaboSPAN analysis to those of classic Fisher's test analysis. We found that MetaboSPAN successfully highlighted several pathways known to be relevant to sarcomas in the literature that were missed by the Fisher's test, including sphingolipid synthesis, pro-inflammatory lipid synthesis, bile acid pathways, and beta oxidation. In conclusion, RaMP-DB enables functional analysis of metabolomic and transcriptomic data, while MetaboSPAN improves the sensitivity of pathway enrichment analysis of metabolites without excessive inflation of false positives. Follow the links below to check out RaMP's website and source code. Please contact the author if you wish to test the MetaboSPAN package before release. Thank you.